Veterinary biotechnology. Chemical synthesis of DNA was devised by Hargobind Khorana, a Nobel Prize winner. Most commonly used type of restriction enzymes are of type 2. Major complement component present in serum is complement 3C3. PCR technique was developed by K.B. Mullis or Carrie B. Mullis. Major DNA polymerase involved in replication in prokaryotes is DNA polymerase 3. Most abundant polysaccharide among living system is cellulose. Recombinant DNA technology is developed by Cohn and Boyer. Remember that corona vaccine by Moderna is, I think, based upon the recombinant DNA technology. Number of asymmetrical carbon atoms in ribose are 2. Semi-conservative replication of DNA was proved by Michelson and Stahl. Protein part of an enzyme is termed as apoenzyme. During replication, the enzyme that prevents torsion by breaking DNA strand is topoisomerase. Topoisomerase prevents torsion by breaking DNA strands. Eukaryotic DNA polymerase for mitochondrial DNA replication is DNA protease gamma. Monoclonal antibody technique was developed by Koller and Milstein. The most stable form of DNA and RNA seen under physiological condition is bDNA and aRNA respectively. Type 2 restriction enzymes were discovered by Hamilton Smith in 1970. In prokaryotes, the DNA polymerase having 5' to 3' exonuclease activity is DNA polymerase 1. Concept of transformation was proved by Griffith. During replication of DNA, the separation of double strands is done by DNA helicase enzymes. DNA replication takes place from 5' to 3' direction. Remember the largest number 5, 3, 53. Opposite will be the 35. 35 is the smaller number, so it is the largest number. DNA replication takes place at the largest number or 5 to 3, and remember it as 53 direction. Cracking of GNA code was performed by Nirenberg and Mathiai. Nirenberg has cracked the genetic code. Nucleotide sequence within a gene that is transcribed into RNA but excised before translation is called as introns. Introns are the nucleotide sequence within a gene that are transcribed into RNA but excised before translation. They are not the integral or functional part. Jumping genes of transposons were first reported by Barbara McClintock. One gene, one enzyme, or one gene, one polypeptide hypothesis was proposed by Beadle and Tatum. Opron concept was proposed by Jacob and Monod. The major form of supercoiling found in chromatin is solenoidal. Solenoidal is the major form of supercoiling of chromatin. Phenomena of conjugation was put forth by Ladenburg and Tatum. Histones are rich in amino acid arginine and lysine. Remember that arginine and lysine are the most required amino acids in poultry. First is arginine, then lysine, methionine, tryptophan, tyrosine, and threonine. These are the required amino acids, and arginine and lysine are in histones. The first two limiting amino acids are within the histones. Wobble hypothesis was proposed by Francis Crick. A DNA double helix model was also proposed by the Francis Crick, and the Wobble hypothesis was also proposed by Francis Crick. Bacterial DNA is composed in a structure called nucleoid. Transfer RNA is produced by RNA polymerase 3. Chemical method of DNA sequencing was developed by Maxson and Gilbert. Maxson and Gilbert proposed the chemical method of DNA sequencing. Molecular beacons are probes used in detection system for RT-PCR. RT-PCR uses molecular beacons, that are the probes. Reverse transcriptase was first discovered by Tanim and Baltimore discovered the reverse transcriptase. The enzyme employed for amplification of specific genes in PCR technique is Taq polymerase or Thermus aquaticus DNA polymerase, the thermostable enzyme. In agarose gel electrophoresis, movement of protein is based on charge to mass ratio. Phenomenon of transduction was proposed by Zinder and Lederberg. The medium used for selecting myeloma cells in hybridoma technology is HAT medium. HAT medium is the medium used for selecting myeloma cells in hybridoma technology. Amino acid that does not exhibit optical activity is glycine. Glycine do not have the optical activity. In nucleotides, both types of pentoses are in beta furanose form. In alkaline conditions, RNA is rapidly hydrolyzed due to presence of two hydroxide groups. Hinge region of immunoglobulin G is rich in proline. Imidazole group is present in amino acid histidine. Imidazole group is present in amino acid histidine. In SDS page, sodium dodecyl sulfate polygel electrophoresis, the movement of proteins is based on mass. Separation of proteins in isoelectric focusing is based on isoelectric point of the particular protein. The reagent developed by Sanger to identify the amino terminal amino acid is 1-fluoro 2,4-dinitrobenzene. Beta turn is secondary structure of protein. Alpha sheath and alpha helix and beta plated sheaths. Alpha helix is the first primary and beta plated sheaths is the secondary structure of protein. The most abundant amino acid present in collagen is glycine. Hershey and Chase first reported that DNA is genetic material. In reverse competitive inhibition of enzyme reaction, Vmax remains the same but Km increases. Vx same, Km increases in reversible competitive inhibition of enzymatic reaction. Cofactor or glutathione peroxidase is selenium. Selenium is vital as it is the cofactor for many enzymes as glutathione peroxidase. In agarose gel electrophoresis, the DNA is visualized using ethidium bromide. Ethidium bromide is very toxic and carcinogenic, so handle it with care in gel electrophoresis. Metabolic anemia often occurs due to deficiency of folic acid. The prosthetic group present in aminotransferase is pyridoxal phosphate. Reverse transcriptase are present in retroviruses and hepatnavirus. Retrovirus has importance in veterinary and human side. Retrovirus along with hepatnaviruses has the reverse transcriptase. A diploid cell line of human origin is HeLa. HeLa cell line is diploid human cell line. Vero cell lines are obtained from African green monkey. Cell lines are commonly preserved in liquid nitrogen at -196 degrees centigrade. Viruses commonly used for production of vector vaccines are large sized vaccine viruses as fowlpox virus, reovirus and hepatnavirus. Coronavirus cannot be used as viral vector as it is very small in size. The fowlpox, reo, retro, and herpes viruses are large in size, so they are preferred as the vectors for vaccine. Size of prokaryotic cell generally ranges from 1 to 10 microns.